Auerbach, and I will be your science teacher today.
No, I'm Mr.
So yesterday we talked about DNA. If this were a molecule of DNA, what would be wrong with this picture?
It only has one thingy.
It only has one thingy. Can we be more specific about what a thingy is? One
The
strand,
ladder
yes.
of letters.
And so if we were going to have another strand, it would be across from here, right? And it would have
T A T G T T T G C T A G T A T.
T. Okay, so for those of you at home not familiar with our game, DNA always and RNA always have complementary bases, meaning that A always matches up with T. So if there's an A on one strand, there'll be a T on another, and so we'll put an A here and a T here.
Oh.
And then if there's a C on one strand, what goes on the other?
G.
G. And then
six.
T, 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 G.
C.
Uh-oh, what do I do here? C,
C.
A, T, C, A, T,
G.
G.
A.
I kind of screwed up the, okay, A,
T.
and T. Right, so that's a complementary pair of strands, and actually, just really quickly, before we get into the RNA and the protein synthesis, when a DNA strand copies itself, what it does is it separates these out, and it'll build new complementary strands across from the other ones. And so, just really quick, it would, if this one were to do that, we would build a new strand right there with a T here, and an A, and a T, G, T, A, G, C, A, T, G, A, T. And notice how this is exactly the same as this strand used to be. So it's like you take this one out, you use this as a template, you build a new one, you do the same thing here, and you'd have two identical DNA strands. But that's not what we're doing today. But wait, there's more. For only $19.99, you're going to get a free lesson on messenger RNA, which is the molecule mRNA. Who was asking me about that? Somebody was just asking me about mRNA. Messenger RNA is the molecule that goes out into the ribosomes and makes protein. Yes? Because that is the job of the ribosome, and it does so with instructions from the DNA. And so, I'll use the blue marker. That probably shows up pretty well on camera. And so what happens is, in the nucleus, messenger RNA gets made onto the template strand, and I'm going to erase the other strand to get it out of the way, but that would still be there. It's just that it would be opened up, and you would make your messenger RNA in exactly the same way, except that what's different about messenger RNA?
It has U's.
Right, so DNA is C, T, A, and G, and it's double-stranded. RNA is different, messenger RNA is different. It still has C, it still has A, and it still has G, but instead there's letter U, it's the place of T, and that allows the cell to be able to tell the difference in case there's any question. Then the U makes it clear that this is RNA and not DNA that's in the wrong place or something. And so that is what we'll do here. When we build this RNA strand, normally we would have had a T over here, right? Well, we would put a U. U stands for uracil, by the way. Okay, so this process is called transcription, when we transcribe the DNA onto the RNA. So what comes next here? Just a little practice.
A. U. G.
U.
U.
U. G.
C. U.
You meant to say A, I'm sure.
U.
I guess I'm really being responsive to you guys. Huh? I just write whatever I want after you say things.
U.
Yes. Okay, so now we have our messenger RNA, and we're going to send that out into the cell in order to get translated. So you have transcription, which is when you write it onto the RNA strand, and then you have translation. And translation looks like this, sort of. The ribosome is kind of like a hamburger. Una hamburguesa, for our Spanish speakers. Okay, So,
Your mixture was right.
what's that?
So the mixture was right because all the gels up in the arms.
Okay, and so what happens is the ribosome has a top and a bottom, and the messenger RNA is going to feed through. And the letters are going to get read. Quincy, I'm going to send you a link to a video that shows this part of it a little more clearly that I'm going to show them in a minute. But
Why don't we get to see it?
what?
Why don't we get to see it?
The part about where I'm going to show it to them in just a minute. I said that.
Oh.
Them was you guys.
Oh.
Everything that he does is awesome.
Anyway, so here we have our strand. I'll put the letters on here just so you can see that I haven't changed anything. So A U G U U U G C A U G A.
We have to write all the letters.
You don't have to. No, I mean you can write some of them. Write the first one, and then so actually. This would be a good place to pause. I'm going to pause this because we have to know where it starts, and I want to pass out something to the class before we do this. Oh, if you don't mind, yeah, just hit the red button. Thank you. Okay, we're back, kids, and now we have our translation sheet, and we're going to use it to translate this RNA message into amino acids. And what happens is, as the message goes, as the RNA gets fed through in this direction, it gets read 3 letters at a time. Every 3 letters is called a codon.
I'm so scared about the final.
Codon. Don't be scared about the
I'm
fun.
going to.
And I don't want to talk about it right now because we're talking about this. Every 3 letters is called a codon, and it gets read by the ribosome as the RNA feeds through. And every 3 that it reads stands for 1 amino acid, which gets added to a chain here, amino acid, and then the next one, and then the next one. And it makes a little chain that will ultimately be the protein that's coded in this piece of RNA. And the way that you do it is you read the 3-letter codes with this box that you have in front of you. We're going to use this box here, which is basically just another version of what you have in front of you, except you'll notice that it says first letter, second letter, and third letter. Why don't you do the same thing? See how it says first letter, second letter, third letter? On the left side of the box, why don't you do that on yours? Write a 1, and a 2 on the top, and then a 3 on the right-hand side. So you'll have the same thing. And this is really how you read 3 letters at a time to get the amino acid. And so each of these boxes shows one of your 20 amino acids. That's these 3-letter things, except for this one that says start, and these 2 that say stop, and this one that says stop. Guess what those tell you?
Stop.
That's right.
No.
They say stop, and then the other one tells you when to start. And in fact, that's the one that the ribosome always looks for first, AUG. That's because when they get transcribed, maybe a few too many letters get transcribed, and so it'll always look for that AUG, and that's your starting point. So we had, what was it, A, U, A, U, G, U, U, U,
We don't have start on ours. We like right there.
G, C, A. Oh yeah, MET. Yeah, that MET under AUG is start. Sorry about that. C, A, no, U, 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 G, C, A, U, G, A. So yes, this one right here, the AUG is always start. And so the cell is going to, the ribosome is going to look for that, and it's going to disregard anything that comes before that, like this first one. And so it'll find that, and then, so it'll read these 3 at a time. Oops. And then it'll stop. So the first one, AUG, the way you read this, is always the same. You start with your first letter, which in this case is A, then the second letter, then the third letter. So the first one we're looking for is AUG. We're going to look for what that means. And the way you read this, you start on the left-hand side where it says first letter, or where you have a number 1, and you see that you have 4 letters to choose from here. Yes. And so this first letter, A, tells you that it's in this row going across here. Some of these. So that tells you that it's in this row. And then the second letter, U, tells you that it's also in this column, which means it's where this row and this column intersect, which is this square here. And then the very last one, G, notice how when we go over here, where it says third letter, we have U, C, A, and G. So AUG, that's telling us that the one we're looking for is in this 4th row. And so AUG is MET, which is methionine, MET. And so that becomes our first amino acid on the chain. Uh-huh.
So there's like 1 letter for each, then the third letter is another letter. Does that make sense?
1 letter for each big row, and then the third letter has every letter in the same row. It's because it's getting more specific, I guess. And sometimes you'll see that, like,
But some of these are the same.
right, some of these you'll see that every single one is going to be the same thing, so it doesn't even matter what the third letter is. The third letter could be anything. Other ones have a couple of amino acids. This one has 2 amino acids and a stop command. So
So then how does the cell know what it is if it creates the same thing?
it's redundancy, so multiple ones can stand for the same amino acid. It's just a system of redundancy. There's 64 possible combinations of the 4 letters in 3 at a time,
So some of the combinations are
and there's only
useless.
20 amino acids. I would say redundant is a better word, meaning they both mean the same thing.
But if it mattered, then maybe they make a different thing.
Well, I think it helps for it to be a little loose. And when we think about mutations, like for example, if you have something that's, letter C, and then a mutation knocks one of these out and changes it from one to the other, you haven't changed the amino acid, so it's actually a little bit of cushion against mutation, like a safety net. Exactly. So okay, so by the way, is this part on camera over here?
Let's throw our way.
Yeah. So here's our ribosome reading our thing. We just did AUG, and that tells us that it was methionine, MET. And so a methionine will be put on the top of the ribosome, and then it'll read the next one, which is UUU. And that will, whatever amino acid that stands for, will get added here.
What if it added, wouldn't the met be the end of the chain? Does it happen first?
You're right, it probably does get added from the bottom up. Yes. It's harder to draw, but yes.
That would make me put something on
It does make sense, you're right. In fact, for just 1 second, let's look at, since we have it on screen and Quincy can see it also, this is what it really sort of looks like as it's getting built. This is called a transfer RNA, and it's going to come in here, and it's going to have a matching anticodon to whatever's here. In this case, this is AGG. That's CCA. That doesn't match, so it's going to leave. we were looking for one that says what? UCC, right? Here it comes. And so,
It's like trial and error.
well, sort of, yeah, they're sort of swimming all over the place in a way. And so, right, then what's going to happen is, this is all going to move over. This is going to get added to this chain, as you correctly said, at the beginning, not at the end, in just a second. But there's an energy step that comes in, and then that gets put on. And then we'll have the next one.
What is that thing?
DDP provides ATP energy.
What are the things that are called it?
These are called transfer RNAs, and what they are.
We haven't talked about that.
No, and honestly, I kind of was going to leave that alone. But what they are is they're these little other kinds of RNA that have anticodons for every possible amino acid, and then they have the amino acids attached to them. So when the right one comes here, finds itself sticking, then it'll get attached to the chain, and then the transfer RNA leads. I'm sort of stopping short of having you guys know that because I feel like that's it's fine. It's fine when you do this in college, but right now I just need you to know that this chain gets built by these things getting added on based on what's written here. So let's go back. Okay, so let's do. So we still don't understand. You're probably not the only one. Don't feel bad. So let's do the next one. The next one in the chain is UUU. UUU. Okay, now don't shout it out as soon as you see it. Let's take time to go through this. So again, first letter, 1, 2, 3. That's what they mean. First, second, and third letter. So the first letter, all the letters are the same in this case. The first letter is U. But what this first U does is it tells us that the amino acid we're looking for is one of the ones that's listed in this row. So now we're going to look at the second letter, which is also U. And so we'll go to the second letter part, which means that you find the letter you're looking for. In this case, it's U. Which it is, yeah, or oh gosh, what somebody else said, another good analogy was made. Yeah, it just takes a few times through and you can get it. So now we've isolated it down to this box, and then you can either just read all 4 of them here. I mean, really, you can see that UUU is here, and then it's phenylalanine is PHE. Or you can go to the third letter side and say, okay, here's my U. That must mean it's in the top row.
Yeah, ours don't have all the letters, so it's not nice.
Yeah, this is very good.
What do you mean? Yeah, dude.
It just is,
But like yours has next to the element of the amino acid thingy, it has all the letters. Ours doesn't have that, it just has the
Oh, I see. Yeah. I do see what you mean. Right. Yes, this one is much better. I'll try to print this when I put it on your final exam. I'll try to print this. I may not remember. I probably won't, but I'm on camera saying, okay. So, and then let's see. The third one. So, okay. So what would happen is that's phenylalanine, PHE would get added here, and then the next one's going to go through. What's the next one? GCA. So, okay, so GCA.
Should we be writing this down now, or does it
in this chain right here, the one that says 1 and then GCA is number 5, right?
Well, yeah, but don't, let's not, that's a different thing that we're going to be doing next. So GCA, so here's your G for your first letter. That tells us it's this row. Oops, didn't erase this. So G, C, and for the second letter tells us it's this row. And then A tells us it's the second one up. GCA is alanine. And so as the chain grows,
I don't think there was only 4
alanine, is that what I said? Yeah. You thought what?
I thought you said that there was only like 4 different things.
There's 4 letters that can be combined 3 different ways, right? So you could have letter U first, second, or third. You could have letter A first, second, or third, et cetera, et cetera. When you do the probabilities, what's that? it works out to 64 different possible combinations. Okay, so, and then finally, the next one is UGA, which I'll just write in here. UGA. When that one gets read, what does that say?
Stop.
It says stop, right. UGA. So here's your first letter U. And second letter G means it's in this box here. And UGA right there means stop. And what that tells the ribosome is it's all good, we're done, cut the cord, send this protein on to the Golgi apparatus for fixing up for export. Okay, so what you're going to do, ladies and gentlemen, you will notice there is a strand of messenger RNA here. It does not have a start codon, you just start at the first one and you go all the way through. You'll also notice that on the back is a bunch of circles. The circles stand for each of the amino acids, and so the one with number 1 is your first one, the first 3. Second 3, fourth, third 3, fourth 3. Fifth group of 3 is the one under number 5. What you want to do is, let's have the camera on the word again for a second. Whoops, what did I just do that with? Okay. This is all the amino acids in the beef insulin molecule. In other words, the insulin molecule that cows have in their body. And what I recommend that you do, you're going to group them off 3 at a time, because otherwise you're getting confuse yourselves. So right, 1, 2, 3, 4, 5, you might want to number them also. I did this right. 1, 2, 3. The numbers up top are not super accurate. 14, 17, 18, 19, 20. Gaga. And so, anyway, you get the idea. And what you want to do is you want to go through these. And like if we do number 1 is UUU. We already did UUU one time. That was PHE, wasn't it? UUU is PHE. And so you would write, just like Cassie just said, PHE right here. And then you do the second one, third one, fourth, all the way through. Until you get, oh, why did I do that? That was stupid. Until you get done. Okay? Okay, I'll put these back. But
It's good circle.
it should take you about the last 20 minutes here. Okay, you may stop it now. Thank you.